see this. So guys, again, all you need is a pencil and the lab that you have in front of you, but this comes with a bit of explanation. So guys, we are starting into the last, this is crazy, we are starting into the last unit of the year. And guys, understand, just based upon the nature of the kind of students that you are, which is to say good, I know that starting next week, your lives get infinitely more complicated because AP testing starts, right? Typically, you are taking one or more AP tests. Guys, important things to know about that, even though you're not my AP students, final payments for your AP tests are due Friday. Please don't miss that. And then, guys, in addition to that, please be communicating with your AP teachers about times and expectations. Do you bring pencils? Do you bring pens? What calculators are and aren't allowed. There's a lot to just sort of keep track of. So, guys, be sure you're having those conversations. But, guys, here's the scoop. Miss Call and I are well aware that May at Orem High School gets really, really complicated. Timpanogos as well. Guys, things get weird. So, in an effort to help you stay organized, guys, this is it. I went back and I actually fully populated Skyward with everything that we are doing for the rest of the year. So guys, yellow is homework. You'll notice that you've got 4 homework assignments in the next month. Then guys, red or whatever that color is, those are labs. You'll notice that you have as many labs as you do homework assignments. Why is that? Because, guys, we know that May is sort of a tricky period, and frankly, it's easier to make up labs than it's easier to make up homework. The lab makeups are really straightforward because of how Miss Call and I have recorded videos of the labs. So, guys, it's all there. It's all there for you. And so we have 4 labs yet to do this year. And then, guys, I know you can't fully see this. You just have to trust that off the screen on the right is the moles test that everybody should have taken and remediated if you qualified. If that's not the case for you, guys, as a class, we've moved past this. As an individual, you're welcome to come back and take the moles test. You just need to make arrangements. Then, guys, this dude right here is the reactions test. That's the one that you took last time. You can look forward to getting that back next time. And then, guys, this is the test that we're starting the unit for today. It's acids, bases, and chemical equilibrium. You're seeing already that it's an equal blend of homework and labs. And so, guys, this is how we're going to wrap up the year. When all the asterisks are gone, we're done with the year. So, what does this actually look like? And guys, at that point, it is time. How do I do this? I really don't know. Shoot. Let me think. I can't. I'm just going to quit. There we go. Guys, the way that this works is like this. Here's the calendar. So today, down here in the bottom right, guys, we are going to start the unit with a lab. I know that's weird because we usually start units with information and reinforce with labs. We're going to start the unit with a lab. You're going to enjoy it. It's a great little thing. Then, guys, looking forward to May, you are going to find that we bounce between lecture and lab. So the first day back next week is a day of lecture. But we've already done the lab for that content. Then, guys, after that, we're going to talk about reaction rates. Then we're going to come back and do a lab. Now, guys, if you miss the lab over reaction rates, you actually will still do the lab. You won't watch a video of it. You'll do the lab at home. It does require a little bit of shopping. You'll need to go to the grocery store and get Alka-Seltzer and vinegar. It mentions it in the information. But, guys, that is a lab that you'll make up, not by doing the video, but the lab. These dates are actually not correct. I need to get rid of those. This is obviously not due 5/13. Frankly, I copied and pasted these links from last year's website. That is not true. For you, these labs will be due the next class period. Guys, all of our labs will not have 2-day windows on them. All of the labs for the rest of the year will have 1-day windows. Then, guys, we're going to come back and we're actually going to do back-to-back days of lecture. The reason is because we used to do the material from Tuesday and the material from Thursday lumped together in one big day, and we found that that just was not effective. So the Le Chatelier's principle, 10 and 11, and then the KC stuff, 12 and 13. Guys, those are actually things that are shorter days. You'll get your homework done simply because we've taken 1 day of material and split it in half. Then, guys, coming back after the weekend, we're done. You have one more lab to do, and then you have a test to take. Now, guys, this puts us in a weird spot. Here's why. Your grades, and you know what, let me zoom out so I can draw just to make sure you're all seeing this, and I'll switch to red so it stands out. Guys, we've run into a problem, and I'll populate the calendar with this, but the problem is this. Obviously, this lab will be due when I see you for the test, because it's on the test, you'll see. But then, guys, the problem is this. Your grades are due the weekend. They're actually due on the 22nd. So you're going, wait a second, how are my grades due? It gets even worse. Guys, this is a consultation day, and this is a consultation day on our calendars, but grades are already finalized. You do the math. But, guys, what we're going to do is we will allow you to remediate this last test. So we will get this graded quick like a bunny, and then we will post your scores. I will actually do the essentials analysis for you. So if you demonstrated mastery of the essentials, you will get the grade bump or you will get the extra credit without needing to engage on that. I'm not sure if Ms. Call is going to do that, but that's what I'm going to do. And then, guys, you can choose to do remediation simply based upon your test score. We'll talk more about that when we take the test. And then, guys, when we do remediation, it could be here, more likely here. We're still trying to figure this out. But fundamentally, guys, the deal is you'll be taking the essentials test after your grades are due, which means we'll have to do grade changes, which frankly is a huge pain in the butt, but it's better for you. So that's what we're going to do. So, guys, there you go. Calendar, rest of the year. Thoughts or concerns? Okay, go ahead, guys. Take your time. Oh. So fundamentally, you have 4 homework assignments for the next unit. And then, guys, what you would have seen is 4 labs, but in fact, it's only 3, because one of the labs that was not graded up there is actually the lab that you turned in last time, the percent yield lab. So we have 3 more labs in this next unit, 4 total yet to be recorded, because I haven't graded your percent yield labs yet. Yeah, keep going. So the 22nd, is Sunday. But coming back after that, the 20th is being described as just a regular day. Then the 23rd is a consultation day, Spirit Bowl, and senior yearbook distribution. Then Tuesday, the 24th, is senior send-off assembly, general yearbook distribution, and graduation. How does that work relative to remediation? And then, guys, the 25th is a consultation day for sophomores and juniors, again with the understanding that grades were finalized 3 days ago. Then, guys, the 26th, as it shows, is the the 25th, consultation day. Yeah, and then the 26th is the last day of school and Lagoon Day for the seniors, which this year will be after graduation. Guys, if all of this is as confusing as it sounds, please come to the front of my room and shoot. If you care to know this, guys, please come and shoot. Excuse me, come and shoot a picture of the calendar. It's all there. What else, y'all? Are we good? Okay, so guys, do this. Please grab the labs that you have in front of you, and we're going to get started. Now, guys, the way that we're going to get started with this is going to be a little strange because you are accustomed, probably without knowing it, you are accustomed to starting a unit with material and reinforcing with a lab. Guys, we are switching that. We are starting with the lab, and we're going to use this to introduce material. And now you're going, wait, this looks like a day of lecture. We've got a title up on the board, and just by instinct, you're like, oh crap, we should be taking notes. Not true. Guys, do not write anything that comes up on this screen down because we will go over it again when I see you next. Right now, you just need to know some fundamental things. So guys, do this with me. Read the title of your lab. You got it? Do you notice that it matches up with what's on the screen? Okay, so guys, in order to understand what we're doing in lab today, you need, first of all, to understand this term, the term cereal. And guys, please understand that this word cereal does not refer to Captain Crunch. Guys, this word cereal is the same word that refers to serial killers. So, what is the job description of a serial killer? And guys, it is not to kill a lot of people. We shouldn't even be talking about this. But guys, if you kill a lot of people, that makes you a mass murderer. If you want to be a serial killer, this just got even more weird. If you want to be a serial killer, guys, the thing that you have to do is kill a lot of people the same way every time. So guys, the word serial actually means to do the same thing over and over and over. So guys, that's actually what you're going to do in lab today. You are going to do the same thing over and over and over. So the question then becomes, what are you going to do over and over and over? And the answer is dilution. So guys, remind yourself from the moles unit. When we talked about molarity, we talked about dilution. What does it mean to dilute something? To water it down. And guys, that's literally what you're going to do in lab today. You are going to water down solutions serially. So you're going to take a solution and you're going to water it down again, and then the same way again, and the same way again, and in the same way again. But now the question is this: what are you going to water down? And guys, the answer is right here. The answer is acids and bases. So bringing all those ideas together, what you're going to do in lab today is you're going to start with an acid and you're going to start with a base. And you are going to dilute, water those down the same way over and over and over and over and over. Then what are you going to do when you're done? Well, guys, then what you're going to do is you're going to add an indicator to this. Have you guys all done the thing in biology where you spit on that blue piece of paper and it turns pink because your spit is an acid? Guys, that's an indicator. An indicator is simply a substance that changes color based on whether or not it's in an acid or a base. But guys, here in chemistry, we have really sexy indicators, and our indicators don't just turn blue and pink. Our indicators actually change 14 different colors depending on what pH they're in. Then, guys, when you're all done with that, turning your attention back to the lab, guys, you see that balanced equation in the middle of your lab about 3 inches down? Guys, you're going to carry out that reaction. Front page, about 3 inches down. See that reaction? What kind of reaction is it? One product. Synthesis. Guys, that is actually the synthesis reaction that contributes to acid rain. Look at the reactants. Carbon dioxide. Guys, not only is that a greenhouse gas, which is a whole other set of issues, it is also a gas that gets up in the clouds. What are the clouds made of? The other reactant, water. And the carbon dioxide gets up in the clouds, and when it does, it reacts, and it actually forms, as it says there, carbonic acid. That carbonic acid then rains back down to the ground, and it's one of the contributors to acid rain. So guys, what you're going to do in lab then today is you're going to make acid rain. You're going to take some water and you're going to bubble carbon dioxide through it, and you're going to make acid rain in a beaker. Where are you going to get the carbon dioxide? Guys, you all understand this. When you exhale carbon dioxide. You're going to take a straw and you're going to bubble that carbon dioxide through water, and literally you're going to watch that water turn into an acid. So guys, that's the big idea behind the lab. With this said, let's now turn our attention to this. And again, guys, do not write any of this down, but let's talk. So guys, fundamentally, we'll talk more about this later. When you think about acids, you think about scary stuff that makes you wear goggles and don't get it on your skin. But guys, fundamentally, this is the deal. All acids do is give away hydrogen ions. We'll talk more about that later. But when you get an acid burn, what you're actually getting is a hydrogen ion burn. Hydrogen ion is very reactive. It rips our cells apart and it feels really bad. So guys, acids make hydrogen ion, and the more hydrogen ion an acid makes, the stronger the acid. Now guys, acids most of you are familiar with, but bases not so much. So guys, if acids make hydrogen ion, what do bases make? Well guys, the answer is hydroxide ion. So guys, bases are substances that donate hydroxide ion to water. The more hydroxide ion they make, the stronger the base. Do any of you see where this is headed? What happens when you take hydrogen and hydroxide and stick them together? You get water. And guys, maybe you know this. What is the pH of water? Do you know? It's 7. We'll talk in a minute, but guys, water is pH 7, which is exactly in the middle. Why is that? Because guys, water is perfectly an acid because it contains hydrogen, and it's perfectly a base because it contains hydroxide. It's also the most plentiful substance on the planet, which to me does not seem like a coincidence. But as we move forward, guys, water is both an acid and a base because it contains hydrogen and hydroxide. It's H2O. It looks like this. And in water, guys, we have hydrogen that makes it an acid, OH that makes it a base. It's right in the middle. It's perfectly both. Good? All right. So guys, this then leads us to this idea of pH. Be ready to swallow hard. What does the H stand for in pH? Hydrogen. What does the P stand for? You're not going to like the answer. What does the P stand for? Logarithm. I know. You guys have been trained on the south side of third floor to hate logarithms. Guys, logarithms are not that tricky. They're just ways that we talk about really big numbers. So guys, check this out. Imagine that I'm counting and I go like this. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That's called counting arithmetically. Then, guys, if I go like this, 10, 20, 40, 60, 80, 100, that is actually what is called counting multiplicatively. But then, guys, we've got logarithms. If we count logarithmically, it goes like this. 1, 10, and then 100, and then 1,000, and then 10,000, and then 100,000. Guys, all logarithms is counting by powers of 10. That's all there is to it. That's all you need to know about logarithms. It's just counting by powers of 10. If you flip your lab over really quickly, Do not write anything down. But guys, sorry, flip to the back. See question B? What is a logarithmic scale? Don't write anything down. You now know this is simply a scale that goes by powers of 10. What is the significance of this? Let's talk about it right now. So guys, pH is simply a measure of an acid-base strength, but it's a logarithmic scale, so it changes by powers of 10. But you also have heard of the pH scale. Some of you knew that water is neutral with the pH of 7. What is the range for the pH scale? Do you know? 1 to 14. So guys, which end is the acidic end? The 1 or the 14? Do you know? It's the 1. So guys, the lower the pH, the stronger the acid. When you hit pH 7, the concentration of hydrogen and hydroxide are equal. We call that water. And then guys, when we get above pH 7, we've got more hydroxide than we have hydrogen. You're right, pH 7 is neutral, and it's water. But when we get above that, we've now got more hydroxide than hydrogen. So guys, it goes like this. Water's in the middle, hydrogen and hydroxide are equal. It's water. Then if we're down at this end, we've got more hydrogen than hydroxide. If we're at the other end, we've got more hydroxide than hydrogen. So guys, we already have established that a pH scale goes by powers of 10. What does that matter? And it's this. Guys, when we take an acid and move through the pH scale, it actually decreases by powers of 10. So if we have an acid with a pH of 1, and if we then go to pH 2, how many times weaker is 2 than 1? 10 times. But now, guys, if we go to pH 3, that is not 20 times weaker, it is 100. You getting it? And then if we go to pH 4, it's 1,000, 10,000, 100,000, million. So guys, water is 1,000,000 times weaker than an acid with pH 1. But it goes the other way too, y'all. If we go in the other direction, we can do, hold on, come on, baby. We can do this. Bases do the same thing. If you have a base with a pH of 14, that's pretty scary stuff. But guys, if you've got 13, how much weaker is it? 10, and you get the idea, right? Okay, so guys, what we're going to do today is then this. Check this out. You are going to start with an acid with a pH of 1. You are going to start with a base of a pH of 14, and you are going to dilute it serially. You're going to dilute it by powers of 10. So you're going to take this acid, cut it down by 1/10. It's now pH 2. Cut that down by 1/10, pH 3. Cut that down by 1/10, pH 4, and so on until you get to water. Then you're going to start here. You're going to dilute this by tenths, and you're going to make the entire pH scale you are going to make the entire pH scale in a micro well tray. Remember these things? You used it for the precipitation lab. So guys, read along with me. Grab your labs. You'll notice that this procedure wraps around to the back. Guys, this is the most confusing lab you'll ever do in this class. Just telling you now. If you don't follow the instructions and pay close attention, this is not going to work at all. So here's what we're going to do. We're going to read through the steps, and you're going to say to yourself, what the snap, I have no idea what this means. That's okay, because what we're going to do then is I'm going to model for you the steps, and then it will make sense. So guys, first of all, let's make sure you understand the context. This entire lab, sorry, I wasn't quite ready for this. This entire lab is going to be done in a micro well tray. And guys, on these micro well trays, there are letters and there are numbers. They're numbered across the top, 1 through 6. They're lettered along the side, A, B, C, and D. And guys, this is, and actually, oh, wait a second. We're going to use the smaller micro well trays. They go through 12 and they go down through like G. So micro well tray, smaller wells. So guys, read along with me and be ready to underline and highlight stuff. So guys, it says this, step 1, fill a clean beaker with distilled. I would underline distilled water. You can't use tap water because the tap water here in Utah is very, very basic because of the minerals that are in it. Then guys, step 2, get 2 micro tip droppers. Eventually these are going to end up in the trash. Then step 3, rinse your tray and place it against a white background. Guys, next to step number 3, you may want to write down with distilled water. Do not don't use tap water in this lab. Then guys, step number 4, place 9 drops of distilled water in wells D1 through D7. I would underline those, D1, D7. And E1 through E6. Again, I would underline E1 and E6, and then I would circle like 11 times, not E7. You'll see why in a minute. Then guys, moving along, step number 5, you're going to fill well B1 half full with 1 molar HCl. Then guys, you're going to fill well G2 half full with 1 molar NaOH. Guys, at that point, you are all set up and ready to do the lab. So guys, join me at the screen and here's what this looks like. The first thing that you're going to do is you're going to get some distilled water in a beaker and a 50 ml beaker. Guys, small beaker, right? And then what you're going to do is you are going to put 9 drops of water, 9 drops of water, 9 drops of water all the way through D7, yes? Guys, if you lose count of the drops, just suck up the contents of the well, start over. Then guys, along the bottom, 9 drops, not an E7. Yes? Okay. Then guys, the next step is this. You're going to go up to well B1 and you're going to fill that how full with 1 molar HCl? Half. And then you're going to go down to G1, also half full of 1 molar NaOH. The dropper bottles are up front. You guys good there? All right, guys. Now it's time to get cereal. In order to get cereal, you need to realize that all these wells have 9 drops of water in them. Now, reading along, we're going to turn our attention away from the screen for a minute, starting under acids. Using the empty microtip dropper, it's got to be empty, you may want to underline that. Using the empty microtip dropper you used with the water, add 1 drop of 1 molar HCl to well D1, returning the unused acid to well B1, and then flush the dropper with distilled water 3 times, putting the rinse water in the sink. Then guys, step 8, with your flush dropper, draw up the contents of D1, put 1 drop of D1 into D2, then return the remaining solution to D1. Then guys, step 10, drop the contents of D2, put 1 drop of D2 in D3, putting the rest back in D2. Step 12, get cereal. Continue doing this through well D6. Guys, I would encourage you to circle, underline, whatever you need to do, D6. If you touch D7, you've got to start over. So guys, join me at the board and let's explain what this means. What you're going to do is this. You are going to put 1 drop of 1 molar HCl into well D1. Now guys, where are you going to get that 1 molar HCl? B1. And you're going to take 1 drop of this and you're going to put it in there. Now guys, do this with me in your head. 1 drop plus 9 drops is, logarithms, right? You just took 1 molar HCl and you diluted it by a factor of 10. You just did a logarithm with a dropper. Now guys, here's the deal. When you do this, this is no longer 1 molar. It is now 0.1 molar, and that has a pH of 1. Then what you're going to do, guys, watch. You are then going to flush out your dropper 3 times down the drain, and then you're going to suck up the entire contents of D1, and you're going to put 1 drop of it in D2, and you're going to put the rest back. When you put 1 drop of D1 into D2, you just did another logarithm. It's now a tenth less concentrated, so now it's 0.01 molar, and that has a pH of 2. Then you're going to take 1 drop of D2, put it in D3, put the rest back. You just diluted that by a factor of 10. Now it's 0.001 molar, which is pH 3, and so on. Do you see the logarithms going on here? So guys, you're just going to keep going and going and going, but do not touch D7. Because guys, what's already in D7? Water. So guys, here's what you just did. Well D1 is pH 1. Well D2 is pH 2. D3, this will be pH 4, 5, 6. And this is already pH 7 because it's got water in it. Get it? Guess what you're going to do now? Oh, please. So 2 answers. The highest possible acidity for HCl is 12 molar. The pH of that's saturated. You can't make it stronger at room temperature. That's how I buy it. 12 molar hydrochloric acid terrifies me. Its pH is negative 1.2. Something like that. So understand when we say the pH scale goes from 1 to 14, that's biologically. I have sulfuric acid that has a pH ranging towards negative 2. Yeah, it's really scary. And I have bases that have pHs in the range of about 15.5. So yeah, also frightening. So yeah. That's the whole idea. Yeah, that beaker of water is your little source of distilled water that you'll use throughout the lab.
What's dangerous about having pH?
So good question. What's dangerous about 14? I mean, we understand that 1 is scary, right? So in acids, we're scared of the hydrogen. In bases, we're scared of the OH because OH is just as corrosive and crazy as hydrogen. We just don't think about it much because we do more with acids than bases. But a good example is oven cleaner. That stuff you spray in your oven and it eats all the gook out of your oven. That's actually sodium hydroxide with a molarity of about 0.5, 14.5. And it'll mess you up. You don't want that on you. So bases are just as crazy as acids. We just don't think about it as much. You guys good? Okay, so now guys, looking down at part base, bottom of the page, step 13, using a new microtip dropper, I would underline new. Add 10 drops of 1 molar NaOH to well E7, because I would underline 10 in E7, because you're going to miss that. Put the remaining NaOH in well G1, flush the dropper with distilled water 3 times, discard the water down the sink. Now guys, step 14, we're going to get serial in the other direction. With your flush dropper, drop the contents of E7, place 1 drop of E7 in E6, put the rest back. Step 16, drop E6, put 1 drop in E5, put the rest back in E6 and keep going. So guys, join me at the board. So what you're going to do is this. You're going to put 10 drops of NaOH into well E7. There's nothing in there. You haven't diluted anything. It is now 1 molar NaOH. That has a pH of 14. Then you're going to suck this up, put 1 drop in there, put the rest back. That is now 0.1 molar OH, pH 13. Doing that again, 1 drop in there, put the rest back. That's now 0.01 molar, pH 12. And you just keep going. And keep going, pH 11, pH 10, 9, 8, and so on. You get the idea? Okay. Now, guys, what are we going to do? Well, what we've just done is we've now made the complete pH scale, right, from 1 to 14. Flip your lab over, and this is where this gets interesting. What you're going to do then is you're going to come to the front of the room, and you're going to get this vial a microtip dropper that has this weird liquid in it that looks kind of greenish brown. That is what is called universal indicator, and it's truly what the name implies. There are 14 different dyes suspended in alcohol, and you are going to put 1 drop of this in each one of the wells, and each one of those wells should be a different color. Now, guys, understand, it's not radically different colors. It's red bleeding into yellow, I think, into green, so it's more like a rainbow than these really distinct color changes, but you should get a slightly different color moving through a range from 1 to 14. I would encourage you to shoot a picture of it. Feel free to take your phones. Shoot a picture of it if you'd like. But in either case, what you're going to do is this. See your data chart? Down here with the circles, see where it says color? You're going to write down the colors. Be as descriptive as you want, but when you're done, don't throw it out. Then, guys, reading along from there, acid rain. Here's what it says. Fill the 50 milliliter beaker 3/4 full with distilled water. There's already some in there. Fill it 3/4. Then, guys, in step 2, make that 10, not 3 drops, 10 drops. Place 10 drops of universal indicator in the water. Record the color. Why will it not be the color of pH 7? Why is actually, wait, this is distilled water. Never mind, I was thinking tap water. Distilled water will it should be close to the color of our pH 7. If it's not, you're okay. This is where this gets good. With a straw, you are going to blow carbon dioxide through the solution. Guys, as you do, the carbon dioxide reacts with the water and it makes an acid. How are you going to know that this is taking place? Hi. Who? Manaya? Yeah, I got you covered. Thank you.
No, I need her to come with me.
And can you wait 2 minutes?
Okay.
Thank you. Okay, so guys, continuing along, guys, with the straw, you're going to blow carbon dioxide through here. And how are you going to know something's changing? You're going to see the color walk backwards through all the colors up here. How do you know when you're done? When you're lightheaded. So when you're lightheaded or just sick of blowing through the water, record the new color. Then, guys, notice what it says down here, pH. How are you going to know the starting and the ending pH? By comparing with the colors up here. Then you're going to answer these 2 conclusion questions and turn it in next time. You guys good to go? Okay, goggles for sure. Everything that you need is up front. We'll see you next door.